So, respected teachers, sirs and madam, and dear students, you all are welcome in this science dialogue series. This is non-profit and non-paid exercise. You all are welcome. If you think you have some expertise, you can also deliver. But we have started with top scholars from the world, different institutes of the world. And I'm happy to introduce Dr. Parth Joshi, today's speaker. And I feel happy that he's Gujarati and he did his PhD and postdoc from world's top second rank institute, that is Max Planck Institute. Nowadays, he's working at Shankar Netralaya, Bangalore. So with this brief introduction, without wasting time, I always try to stick on time because I have seen webinars generally start after 10-15 minutes from the scheduled time. So I don't prefer that. I always stick to the time. Whatever number of participants are joining or not joining doesn't matter. क्योंकि हम समझते हैं कि सारे लोग नरेंद्र मोदी और सारे लोग नोबेल लॉरिएट नहीं बन सकते, भीड़ कभी सफल नहीं हो सकता है. Those who are interested, they will attend, they will join. That's why if people are not attending the last slide, so they will also not get certificates. Usually we don't give certificate, but if you will attend completely and share your feedback, then after few lectures, few sessions, we will decide to give you certificate also. But my motto is to disseminate knowledge, quality knowledge, not only knowledge, but quality knowledge, so that our all knowledge pool of society and nation will improve and increase. That's why we have requested several scholars from different corners. We are in connect with so many. You all know that so many previous lectures. One speaker was from Oxford. Today he is from Max Planck. So we all, and this is not one way lecture. Sir Rakesh Patel, sir, please mute your mic.
Yes, sir.
Yes, sir. So you can interact, you can ask question anytime, but I prefer that you should ask after this, but after lecture, but you can interact, you can ask question in between also, because our speaker is not like a conference. They will deliver 20-minute lecture and then they will take question. No, I generally prefer young scholars who can take your all queries, and you can interrupt him or her anytime, so that you will not pause, your brain will not pause. Suppose once we get stuck on any terminology or anything, so our brain will not accept next terminology. So whenever we conduct the course, we try to interact with the participants. No one is in mute condition. All are in that automated condition. You can unmute and speak. So with this brief introduction, and I really thankful to Dr. Parth Joshi for sparing his time. Everybody should take should spare his time for improvement of our nation and our society. So his contribution will be duly acknowledged, and we all are grateful to Dr. Parth Joshi. Please continue, Parth.
Okay, thank you so much, sir, for this nice introduction. So as you mentioned that this is an interactive session, and actually I would also I would be happy if you can ask me questions in between, so that we can communicate better. So because this is a very new topic, I am guessing, and so that's why if you feel that something is not, if you are not understanding anything, please feel free to, ask. Okay. So I will start now. Okay, sir. So my presentation is visible, right?
Yes, it's visible, and you are audible also, clearly audible.
Okay. So, okay. So thank you, and I welcome everyone for my talk. So today I will be talking about long non-coding RNAs. So since very beginning, so since my PhD, I was more interested to you know about the RNA world, and especially the non-coding RNA part, and that is how I came across this topic. and I wanted to know how these long non-coding RNAs, how they are actually regulating the biological processes. So, um, basically, what we have learned or at least what I had learned during my college and during my university was that the central dogma of life is only DNA that gives information, to transcribe into RNA, and RNA gets translated into proteins. This is the only thing that I had learned. But apart from this, there was a very nice book that I read during my college that was titled as the RNA World. And in this book, RNA all the biological processes and how this RNA can be used as diagnostic markers or for treatment. Okay, so that is how I came across this long non-coding RNA field. So I will just give you a very brief introduction. So how this RNA? So basically, in the 80s, there was the discovery of the double helix and the nuclein, but no one was actually talking about RNAs, correct? And so it was only in 1991 that someone first time discovered long non-coding RNA that was called the Exist. So Exist is a long non-coding RNA that is basically required to silence the X chromosome. So you know that in males there is only one X chromosome, in females there are two X chromosomes. But in order to compensate the dosage, in females one of the X chromosome is silenced, and that whole phenomenon is performed by a long non-coding RNA and not a protein. So this was very interesting. Eh, So after 91, people started focusing more on this link RNA part. And around 98 or 2000, the first hints of link RNAs being present in a larger quantity in the genome came out, and especially in 2002 when first time someone said that the entire genome is transcribed. Before that, people thought only proteins are the main components of a cell, but no. After 2002, after RNA sequencing and all these technologies were developed, we realized that it's the whole genome that is transcribed, correct? And in 2012, for the first time, people said that yes. So the researchers, so there was a very nice paper from the ENCODE project. So this is the encyclopedia of DNA elements. So for the first time, people saw that this whole genome actually codes for non-coding RNAs. And this is how the journey of this link RNAs started. So, okay, sorry. Yeah. Okay. So what was known before that only 1 or 2% of the whole genome is actually translated into proteins. The rest of the genome, you would have heard the term called the dark matter or junk DNA. So people thought that the 98% of the genome is junk, but that was not correct. After sequencing technologies came up, people actually confirmed that this is not junk DNA, but it actually codes for two type of non-coding RNAs. The first type of non-coding RNAs are housekeeping non-coding RNAs, which we all know that include ribosomal RNA, transfer RNA, small nuclear RNA, and small nucleolar RNA. So these housekeeping non-coding RNAs actually are required for RNA editing, for the transfer of amino acids or for the ribosomal proteins. But there was another set of non-coding RNAs which is known as regulatory non-coding RNAs. Now this is very important. Regulatory non-coding RNAs is again divided into two parts. One is linear RNAs, second is circular RNAs. Linear RNAs can be further divided into long non-coding RNAs and small non-coding RNAs. We have heard of small non-coding RNAs, which include microRNAs, that is responsible for RNA interference, or small interfering RNAs. Again, we have heard of siRNAs and shRNAs being used for the knockdown of several genes. Correct. So the long non-coding RNA part was mostly untouched, and no one knew that how these link RNAs are actually functioning inside the cell. So today I will talk about link RNAs. Now, first I will tell you what is the definition of link RNA and how can we identify these link RNAs. Then we will move on to the classification. So, link RNAs are transcripts that are transcribed by RNA polymerase 1, 2, or 3. Mostly they are transcribed by RNA polymerase 2, but they can be 1 and 3 also. Transcripts that are longer than 500 nucleotides. This is the most important criteria, but lack coding potential. They cannot code for proteins. Why they cannot code for proteins is due to absence of a reliable open reading frame. If they fulfill these three criteria, then they are called as long non-coding RNAs. Another hallmark of link RNAs is they have active H3K4ME3. I will explain you what this is, and H3K36ME3 modification marks. Now, for any gene to get transcribed by RNA polymerase, it has so the chromatin structure has to open up. You would have heard of two terms, heterochromatin and euchromatin. So whenever polymerase 2 has to transcribe any portion of the genome, the chromatin has to open up. Now, in order to open up the chromatin, these methylation marks are very important. So these methylation marks on lysine 4 and histone 3 and lysine 36 on histone 3 are very important to indicate that this portion of the genome has been opened up for the RNA polymerase to go and bind to the promoter and transcribe that gene. So if any gene body shows active marks for these two uh, 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 so active marks for these two methylation, then it is an indication that the RNA polymerase has been opened up and transcribed the gene.
आपकी आवाज नहीं आ रही है, मुझे नहीं आ रही है, किसी को भी नहीं
सर अभी भी नहीं आ रही।
आ रही है।
नो सर।
आपकी आवाज नहीं आ रही। हेलो, यू आर नॉट ऑडिबल। ये मुझे लग रहा है ईयरफोन लगा लिया। जस्ट मैंने अपना इंटरनेट हेलो हेलो, आपकी आवाज नहीं आ रही। हेलो, आपकी आवाज नहीं आ रही थी कि पूरा स्लाइड आपका नहीं आवाज आया उसका। नहीं नहीं, ये जो है ना 5 मिनट से नहीं आ रहा आपका।
Hello. Right? Okay. Okay, this one, no. ये वाला.
Yes.
Okay, ये clear थी. ये slide.
हाँ हाँ इसको कर दीजिए.
Okay. Uh, yeah. Okay. So was this slide clear?
Now slide is not visible.
This is clear.
Your slide is not visible, na?
अच्छा, कुछ connection problem हो रहा है.
PowerPoint is not visible.
Okay, now it's visible.
No, not still.
Now it's
Okay,
fine.
it is there.
हाँ हाँ हाँ. ये आपने बताया था इसके बाद बोला है ना, उसके बाद आवाज नहीं आ रहा था.
Okay.
हाँ
अब visible है slide.
हाँ 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 विजिबल है इसको.
Okay, आवाज आ रहा है.
हाँ आवाज आ रहा है.
Okay, so this slide was clear or should I explain this
यस
again?
यस. अब इसके बाद कर दीजिए.
Okay, from here.
यस. इसमें भी मिथाइलेशन तक आया था थोड़ा सा, सेकंड जो पॉइंट है उसके
Okay,
बाद.
okay. हाँ हाँ मैं पूरा करता हूँ. Okay, so long non-coding RNAs. पहले हम so first we will focus on the definition, and then we will move on to the classification. So, link RNAs are transcripts that are transcribed by RNA polymerase II that are longer than 500 nucleotides but lack coding potential due to absence of open reading frame. Means any transcript that is longer than 500 nucleotides but they do not have an open reading frame and that cannot be translated into amino acids. They are known as long non-coding RNAs. Now, the two hallmarks of link RNA are H3K4ME3 and H3K36ME3, meaning methylation of lysine 4 on histone 3 and methylation of lysine 36 on histone 3. So basically, whenever RNA polymerase has to transcribe any gene, so it has to be so the chromatin has to be open up. In order to open up the chromatin for the RNA polymerase to actually bind to the genome, it has to undergo these two modifications. Now, if these two modifications are high on any gene body and promoter, that means there are high chances that we will find a link RNA gene there. Now, link RNA promoters and exons are evolutionary conserved, meaning that nature is conserving something other than mRNA and proteins because it is functionally significant. And majority of the link RNAs are enriched in the nucleus. So more than 90% of link RNAs are found in the nucleus, but there are few cases where link RNAs are also present in the cytoplasm, mitochondria, or other cell organelles. Is it clear, everyone?
Yes yes.
Yeah. Okay. Now, in order to fully understand the function of link RNAs, people have classified them into 6 classes. Now, this is very important because if we want to know the function of any link RNA, we have to know where is it located exactly in the genome. Correct? Now, so these 6 classes are as follows. So, first class is the intergenic link RNA. Intergenic link RNA means if a link RNA gene is located in between mRNA gene 1 or any protein coding gene 1 and protein coding gene 2. So, in between any two different mRNA genes, then it is called as an intergenic link RNA. And this class of link RNAs actually comprise the highest number of link RNAs in the genome. Second class is intronic link RNAs, meaning if a link RNA gene is present in between the exons of any protein coding gene, then it is called intronic link RNA. So don't get confused between intergenic and intronic. Intergenic means randomly between any two protein coding genes. Intronic means specifically in the intron of a particular gene. Is everyone clear?
So so then this is called the intronic link RNA gene. And if the link RNA gene is overlapping a protein coding gene, so here the blue one is the protein coding gene, so there are two different exons. So if the link RNA gene overlaps the protein coding gene and is transcribed in the same direction on the same strand, DNA strand, then it is called a sense link RNA. Now, there are cases where this link RNA gene and the protein coding gene have often share the same promoter, meaning they have the same promoter for its transcription. Fourth class is antisense link RNA. If a link RNA gene is overlapping the protein coding gene, but it's on the opposite strand and it's transcribed in the opposite direction, then it is called an antisense link RNA gene. Now, antisense link RNA gene are actually known because they are overlapping the protein coding genes. They are known to modulate the expression of their neighboring genes. So basically, these antisense link RNAs control the expression or the transcription of its neighboring mRNA gene. Fifth class is the divergent class, where if a link RNA gene is transcribed in the opposite direction than the protein coding gene, but they do not overlap, then it is called a divergent link RNA. So as you can see, they are transcribed in opposite directions, but they are not overlapping. So basically, the gap between the two genes is roughly less than 50 base pairs. Now, these link RNAs also are known to alter the expression of their neighboring genes, like antisense link RNAs. And the last is the enhancer link RNAs, meaning if any link RNA gene is situated or is overlapping the enhancer of another protein coding gene, then it is called enhancer link RNAs or E-link RNAs. Basically, these link RNAs modulate the transcription because they are sitting on the enhancer, and we know that enhancer actually regulates the expression of or the transcription of protein coding genes. So these E-link RNAs can regulate the expression of these uh, these protein coding genes. Yeah. Is this clear, the classes? Are there any questions on this?
They will ask whenever they will find
Okay.
difficulties or to repeat it. है ना?
Okay, so I'll continue.
Participants, you may interrupt him anytime. You may ask questions because this is interactive session, not one way. Yes, please.
Sir, in the sixth slide, coming to the intragenic, so I would like to know the gene 1 and the gene 2 are from the same element or the functional, or from gene 1 from one thing and gene 2 from the neighbor. Sixth slide.
So you're talking about the intergenic one, right?
Yes, sir.
Yeah. So intergenic link RNA means these are two different genes.
Okay.
Okay, gene 1 and gene 2. So basically, any link RNA is
आप
present
वो
between
पॉइंटर
two
यूज
genes.
कर सकते हैं. पॉइंटर यहाँ नीचे स्लाइड के इसमें कॉर्नर
Yeah.
में होगा, लेफ्ट कॉर्नर में एक बार देखिए. हाँ ठीक.
So
Yeah.
this link RNA gene is located between two genes, randomly between two genes. Let's say this is, I'm just giving an example. This is
Okay.
PAX6 gene.
Okay.
This is, let's say, HTR gene.
Yes.
And this is a link RNA X. So, these are two different genes.
Okay.
Okay.
Combo between two different genes.
Yes.
Okay, sir. Thank you.
Okay. Yes. So now what is now known that the total number of link RNAs actually are higher than the total proteins that are present in any living organism. So the GenCode human gene, so this is the largest library of link RNAs. They have identified that there are 32,000 link RNA genes that are currently present in the mammalian genome. Now here we have a graph that is showing, you have to see the purple bars. These indicate the total number of link RNAs in different organisms. Now, I am showing you this because um, we have to understand that the total number of link RNAs, if it is more than proteins or mRNAs, then definitely it holds some functional significance. Correct? Now, here, what so this number is only a predictive number. That means the total number of link RNAs in different species is not fully annotated. Correct? Because in RNA, so because people do, hello, because when people do RNA sequencing, depending on
शिल्पी
the depth
मैडम
of
आपका कभी कभी अनम्यूट हो जा रहा है, वो डिस्टर्ब हो जाता है.
this is, hello, unmute here.
नहीं नहीं नहीं, आप कंटिन्यू करिए. वो शिल्पी मैडम का अनम्यूट हो जा रहा है, जिससे डिस्टरबेंस होता है ना.
Okay, okay, sorry. Okay, so I was saying that when we perform RNA sequencing of link RNAs, depending on the depth of RNA sequencing, depth of RNA sequencing means, let's say we perform 90 million reads, or let's say we perform 200 million reads. The number of link RNAs that we can detect on both these two different reads will be different. So, link RNAs identified from 90 million read depth will be less than from 200 million read depth. So, due to this discrepancy, no one till date has ever figured out the exact number of link RNAs present in different organisms. So, this is the biggest challenge today that we are facing in this field. Uh,
So
so
even
now,
we are not knowing number of exact protein coding genes also till date.
yes,
It's varying from 21,000 to 25,000.
yes, that's correct. Yeah.
Yes.
So now what I have done, I will also show some of my data so that you can better relate how we can actually detect link RNAs, how they look like, how they're expressed. So I was working on mouse models. So I will show you how the expression pattern is of a particular link RNA in a mouse model, and what can we do to actually understand the function of that link RNA. So the workflow to understand any link RNA. So first of all, we have to find how many link RNAs are present in any living organism. So there are different methodologies to do that. So one is expression profiling. You do sequencing like microarrays. You can do cDNA cloning, chip sequencing, where you have a protein of interest and then you pull down that to identify the partners, the binding partners. Or there are some bioinformatic approaches. Using any of this method, you can actually detect link RNAs. What I have used is RNA sequencing, that I will show you now. I have used RNA sequencing first to identify how many link RNAs are present in a mouse. Then, next, what I did was I performed qRT-PCR. So this is important because from RNA sequencing, sometimes because the technology is not so sound, not so fully functionally what do you say it so RNA sequencing a lot of times can give a lot of background noise, so random repeats it can give. So in order to validate that whatever we are seeing in RNA sequencing, is it a correct transcript of link RNA or it is a background noise, we have to perform qPCR. So qPCR we perform to validate, yes, that the link RNA that we have found is a genuine link RNA. Then you understand where it is located. So what is the expression pattern of the link RNA? Is it expressed in the brain? Is it expressed in the kidney, eye, liver, where? Right? So we do functional annotation of where that particular link RNA is expressed. Last step is to understand the function of that link RNA, that we do either by transgenics. We can perform knockdown assays, knockout assays, or we can use RNA interference to finally knock down that gene and see what is the outcome or what is the phenotype. So what I did was so this is a mouse embryo. E14.5 means it's still under embryonic stage at 14.5 developmental stage. So what we did was we took this mouse embryo we isolated RNA and performed RNA sequencing, total RNA sequencing from the total RNA that we obtained. Then after that, we mapped all the data, all the bioinformatic data to the genome. We also used now, because this is a new topic and we do not know the exact number of link RNAs, we have to use some public databases like UCSC Genome Browser or Ensemble or Reference Sequencing Database, which are already there on the internet, to see how many of our link RNAs actually overlap with already published data. This is important because this will filter out a lot of background reads that we get from RNA sequencing. Now we are not interested in protein coding genes, so we remove that. We also remove small non-coding RNAs that I mentioned in the introduction, like rRNA, tRNA, small nuclear RNA. We removed all of that because we need only link RNAs. Next, we select only multi-exonic transcripts. This is optional. This we did because we wanted to be sure that the link RNA is having different exons. And then we obtain a final set of link RNAs. So what we observed that only in mouse and only at one particular developmental stage, there are 7,000 link RNAs that we detected. And I mentioned about the classes before. So you can see the number of different link RNAs at each different individual class. And I mentioned that the intergenic class is the highest among all the classes. So we can see that there are roughly 3,000 intergenic link RNAs that are present in the mouse gene. So next, we wanted to validate. So what we did was we performed qPCR. So qPCR is like a regular qPCR when we want to detect mRNAs. So basically, when we performed the qPCR of different tissues from the mouse, we saw that, okay, here E stands for embryonic. So until embryonic kidney, this is the embryonic stage, and AD means adult. So after the mouse has born, and once it becomes an adult, we look the expression in different tissues. So P56 means the developmental stage again. So what we saw, that this link RNA, this was the link RNA that I was working on, and it is called GM. So it's a number that we gave because there is no specific name, because no one has ever worked on this before. So what we saw, that the highest expression of this link RNA is in the brain. You can see the relative expression of this gene at in the embryo is in the brain. There is very less expression in the eye. Not There is no expression present in other tissues. In the adult brain, there is expression, but it's less compared to the embryos. But also, the expression is present in adult spleen. So this is just to validate that in which tissues is the expression of this link RNA present. Now, the third step, as I mentioned here, was to see, the expression pattern of a link RNA in an embryonic tissue where it is expressed. So we performed a technique called as RNA in situ hybridization. Now I will briefly explain this technique, but I will be happy if anyone wants to understand this in detail. So basically here, okay, like how the RNA is expressed in any tissue. If we want to detect that, we used a chromogen based technique. So basically, when there is a RNA, this RNA will be detected by an antibody, and this antibody will be colorimetrically, that means using this blue color, it will be amplified. So when we are using enzymatic substrate on this tissue, only where the RNA is present and only where the antibody is bound, that will show a blue color. This is in very simple terms. So here, this is a cross section of a mouse embryo, and what you can see, so this portion is the brain. Sir, you can see my pointer, right?
Yes, yes, yes. It's
Okay.
like immunohistochemistry, na?
Yes, correct. It's like immunohistochemistry, but we are targeting the RNA here.
Yes, not this protein.
Yes. So basically, so this is the brain of a embryonic mice, correct? This is the spinal cord. This is the liver. this is the lung and other organs. But apart from this, what I'm interested here is you have to focus on the blue color. Wherever there is blue color, there the link RNA is present. So this is all in white. So there is no link RNA present in other tissues. So I've zoomed in here. You can see that in the cortex, right? In the cortex, only in these layers, there are two layers, cortical plate and the intermediate zone. There are few dots which are blue. So that means the RNA is expressed there. In the eye, so this is the retina. And correct, so in the retina is there. This is the lens. It's present. So it's showing blue color. So these are the ganglions. Ganglions are a part of the nervous system which transmit the information. So it's blue, correct? And in the kidney, in the kidney there are few blue dots, and also in the limbs. So these are the foot of the mice. So it's blue. So basically, this shows where exactly the RNA is present. Okay. Now, once we know the whole expression pattern and where it is expressed, we have to understand its function. So how do we understand its function? So here what I did was, so here I want to knock out this link RNA. In order to knock out the link RNA, what I did, so this is the gene, link RNA gene. So exon 1, exon 2, exon 3, correct? Please don't focus on the top line of, just focus on this one. So here I wanted to terminate the transcription of this gene. So what I did was, now how do we terminate the
आपका
transcription? We
मार्क
all
का पॉइंटर नहीं दिख रहा है.
हाँ, क्या दिख रहा है अब?
हाँ, अब दिख रहा है.
हाँ. So now, okay, if I zoom, I cannot. Okay. So now, in order to terminate the transcription, what we did was, we know that a transcription will stop only when there is a poly A signal that is present in the genome, right? So we decided that instead of the 3' end of the RNA, we will put the poly A sequence immediately in the exon 1 or after the exon 1. Correct. Now, this approach is basically used when you do not want any gene to be transcribed in its entire length. If you want to stop the transcription immediately after the first exon, you can insert poly A sequence. So this is called the transcription termination sequence immediately at the exon 1, and the RNA polymerase will not be able to transcribe this gene at all. In this way, we are sure that there is no link RNA being transcribed at all. And how are we sure? We performed qPCR first of all. So you can see this is the wild type where there is no poly A signal. The expression of our link RNA is present. So this is a name that we gave to the knockout link RNA, and you can see that there is no expression. That means this is a very successful knockout of the link RNA. And we also confirm this by using in situ hybridization. Here again, you can see the blue means the link RNA is present, but white means when the knockout in the bottom panel, you can see that there is no blue signal that is present. So that means we have completely knocked out the link RNA. And this is how we actually study the function of a link RNA. Now this work is still in progress, so I will not continue this further. But I want to move on to so the next step of link RNA is like, how do we know that what is the link RNA doing in the cell? So there are several mechanisms. Now I told you that most of the link RNAs are present in the nucleus. So in the nucleus, what do they do? So there are several molecular mechanisms that have been proposed by different scientists. And first one is that is the link RNA. So the gray one is the link RNA. They can bind with the polycomb repressive complex. So polycomb repressive complex is a protein that deposits methylation marks. Now this is done to actually repress any gene in the genome. Now if you want to inhibit any gene, so a link RNA can bind to this complex and it can repress the transcription of other genes. And this is one of the functions. Second function is link RNAs can bind with transcription factors. this is the most important, I think, function of link RNA, that link RNAs bind with transcription factors to form RNA transcription factor complex, which can either activate or it can inhibit the expression of other genes in the genome. Basically, what I'm trying to tell you here is that proteins or transcription factors cannot act on their own. They need a support system, and that support system was not identified until the link RNAs came into picture. Now we know that the support system by which the proteins act are link RNAs. So basically, they are the backbone of any molecular mechanism that needs to be carried out in the cell. I am showing you two more examples, and then I will move on to the cytoplasmic functions. So here, link RNAs can also regulate splicing. So we know that the introns have to be removed after any gene is is transcribed. So basically, link RNAs can act as molecules which associate with splicing factors, and they can do the splicing of different exons. Or link RNAs can also compete with microRNAs. So here, this is important because for the first time, people showed that there can be RNA interactions. Like RNA can interact with other RNA. So link RNA can interact with other microRNA, and that can influence the expression of different protein coding genes in the cytoplasm. What so now we have to understand that because these do not code for proteins, right? So majority of them, they reside in the nucleus. Some of them, they go out into the cytoplasm, so they shuttle between nucleus and cytoplasm and the nucleus. So when they shuttle to the cytoplasm, they actually regulate the stability of other mRNAs. We know that mRNA translation happens in the cytoplasm, right? In order to stabilize that mRNA, these link RNAs move out into the cytoplasm and stabilize the mRNAs so that they can actually form better proteins or amino acids. Or the other function of link RNA is they can associate with inducing RNA inducing silencing complexes like RNAi pathways, and basically they can influence the silencing of other protein coding genes. Now there are several mechanisms of link RNAs, so it's not like that link RNAs have only one or two specific mechanisms. They have multiple roles basically, depending on which cell you are looking, or which tissue you are looking, or which disease you are looking. The expression of the link RNAs can be different. So that is the whole idea of this.